Hi everyone, my name's Tanmay and I'm a PhD student at the University of Cambridge. I study brood parasites and hosts, specifically egg mimicry in the cuckoo finch, and this is shown in the picture on the left. The inner circle of eggs are cuckoo finch eggs. These are good mimics of the outer circle, which are eggs of one of the cuckoo finch's hosts, the tawny-flanked prinia. In the middle is a cuckoo finch chick towering over its host foster siblings, and on the right are an adult cuckoo finch and a tawny-flanked prinia. Today I'm going to talk about the very complex patterns on prinia and cuckoo finch eggs. These complex signatures of identity help hosts to identify their own eggs, and cuckoo finch forgeries have evolved to trick the host into accepting a parasite egg. And I did this work with Andrei, Apostol, Chris Town, and Claire Spottiswoode. So why should we focus on complexity? Well, it's an under-researched subject. Biological complexity is difficult to measure, so it's almost never been quantified. For instance, which of these patterns is the most complex? A has more components, but they're all the same. B has fewer components, but has more variation, and they're randomly distributed. And C has the most types of components, but the pattern is predictable. The point here is that it's hard to quantify complexity objectively. Indeed, how complex a pattern is depends on how the receiver perceives it. And this is a problem, since complex patterns are ubiquitous in biology, from patterns on animals to patterns on eggshells. How might complexity be processed by a receiver? Perhaps it could differentiate between patterns based on the absolute differences in complexity between them. However, an alternative is Weber's law of proportional processing, which I'll illustrate here with an example. Imagine that you wanted to steal a cookie from a plate of cookies without anyone noticing. You could either steal one from a plate of 2, as shown on the left, or a plate of many, as shown on the right. I hope you'll agree with me that you'd steal one from the plate of many cookies because this would be less noticeable. Now this is odd, because in either case the absolute change in the number of cookies would be the same. It would be 1 cookie. But in fact, it's the relative difference that's important. The relative difference is much larger when there are only 2 cookies, in which case it's a half, than when there are 100 cookies, in which case it's a hundredth. A hundredth is a very small relative difference, so it's much less noticeable. Weber's law states that when receivers discriminate between stimuli, relative rather than absolute differences between stimuli are processed. The cookie example demonstrates that it's easier to detect a change or a difference when stimulus magnitudes are small, which is a corollary of Weber's law. So let's apply Weber's law to a biological scenario. If you're a host of a brood parasite, identifying the odd egg out here is easy. The egg on the right has one more spot than the others. But when the number of spots is larger, it's much more difficult to identify the egg with an extra spot. It's the one on the right again. This illustrates Weber's law. Discrimination is easier at lower stimulus magnitudes. And if we apply the same argument to complexity rather than number of spots, assuming that hosts discriminate between eggs based on relative differences in complexity, this would mean that hosts should do better at discriminating between less complex eggs than between more complex eggs. And this can lead to some hypotheses about egg signature evolution. Weber's law states that it's easier to discriminate when stimulus magnitudes are small, so hosts should therefore evolve towards lower complexity. By contrast, it's been traditionally thought that hosts should benefit from complex egg signatures because these are difficult to forge, and this predicts that hosts evolve greater complexity, unlike Weber's law. But there's an additional nuance to these seemingly conflicting hypotheses. If host eggs are more complex than parasite eggs, then even under Weber's law, hosts shouldn't evolve towards reduced complexity. This is because hosts would be evolving to look more similar to parasite eggs, which evolution shouldn't favour. And we might even hypothesize that cuckoo finch eggs might be less complex than prinias. Unlike prinias, the most recent ancestor of cuckoo finches, with their closest relatives, probably had plain white eggs, which are not very complex. In this case, cuckoo finches should evolve greater complexity. So we can test these hypotheses with egg rejection experiments. In the field, I would swap a host egg, in this case from a clutch of 4, for an experimental conspecific egg, the bottom right egg. Then I'd photograph the clutch, and then I'd check whether hosts rejected the experimental egg, as this video graph shows. However, we're still left with the problem of how pattern complexity can be quantified in a receiver-appropriate manner, and this is where my collaborator Andrei comes in, who used computer vision techniques to do just that. First, we extracted features or pattern markings using a tool called Nature Pattern Match, and these are the red arrows on the egg image. We then extracted measures that might contribute to complexity, including the number of markings, how variable markings are, and the number of clusters of markings according to different clustering algorithms. We then used an optimization algorithm with training and validation to weight these measures optimally to predict egg rejection. Essentially, this means that complexity is quantified such that the absolute complexity difference between host and experimental egg predicts rejection as well as it can, given the measures that we extracted. And we avoided overfitting by repeating this process on different subsets of the data. And here's what we get: an egg of low complexity on the left and one of high complexity on the right. And this seems a pretty good match to a human eye view of which eggs are more complex and which are less complex. So we optimized the complexity metric such that absolute complexity difference between host and experimental eggs predicted egg rejection. But interestingly, using various methods that I won't go into here, we find that relative complexity difference is a better predictor of rejection than absolute complexity difference, despite the measure having been optimized for absolute complexity difference. This graph shows that as relative complexity difference increases, the probability of egg rejection also increases. The points are individual experiments. On the y-axis, a 1 means the egg was rejected, and a 0 means the egg was accepted. And this result supports Weber's law. It means that for a given difference between host and parasite, hosts with less complex egg signatures should do better. So here, Weber's law suggests that hosts might benefit from less complex signatures. But recall that if parasites are less complex than hosts, hosts shouldn't evolve reduced complexity, since that would make them more similar to parasitic eggs. And indeed, we do find that host eggs are more complex than parasitic eggs, as the box plot shows. The egg images are cuckoo-finches of high and low complexity, and prinias of high, medium, and low complexity from top to bottom, with the numbers corresponding to points on the box plot. And this result would predict that actually hosts shouldn't evolve towards less complex egg signatures. In fact, parasites should evolve greater complexity over time, becoming more similar to their hosts. And that's exactly what we find. Using historical egg collection, we find that over a 50-year period, cuckoo-finch eggs have evolved significantly greater complexity. The box plot on the left illustrates this. Current cuckoo-finch eggs are more complex than historical cuckoo-finch eggs. The box plot on the right shows that over the same period, prinia complexity hasn't changed. And this is exactly what we predict from prinias being more complex than cuckoo-finches. So overall, I've shown that while complexity is difficult, perhaps impossible, to define objectively, we can quantify it in a receiver-appropriate manner. This sort of biologically relevant method could be used to quantify complexity in all sorts of biological patterns. I also showed that prinias process complexity according to Weber's law. And while this might suggest they should decline in complexity over time, they don't, probably because cuckoo-finch eggs are less complex than prinia eggs. And as predicted by this, cuckoo-finches have evolved increased complexity in egg pattern over the last few decades. I hope this study encourages you to consider measuring complexity in your own study systems and to quantify it in a biologically relevant way. And with that, I'd like to thank my co-authors, Andrei, Chris, and Claire, and also thank Tony and Dom for help with statistics. I'd also like to thank many people in Zambia where I carried out the fieldwork and the University of Cambridge for funding. And finally, thank you all for listening, and I hope you enjoyed this picture of a very grumpy-looking cuckoo-finch chick from my fieldwork in Zambia.